I present FluoGuide, and very happy to be here live for a long period of time since last time. So FluoGuide, we maximize surgical outcome by making cancer fluorescence. And we do that, I will explain in a second, very good result we just announced here for a few weeks ago. This is a disclaimer, so please make your own assessment. So this is the problem in a nutshell. I mean, while the problem for the surgeon is that cancer recur locally in half of the patient approximately. It's not because the surgeon is poor in any way, it's just because it's very hard to see where the cancers stop and where the normal tissues start. And on the image there on the left side, you see a normal brain tissue, it appears. Underneath there's in fact a cancer, but you cannot see it, and the neurosurgeon have the same problem. On the right side, we have switched on the light, and you can see very clearly that the green light marks the cancer cells, and that is a cancer underneath. This is how it works. Our product is a freeze-dried powder. It's reconstituted. It's put into to the blood vessels of the patient before surgery, and then it will stick to the cancer, and then it will be disappearing from the body. Then during surgery, when the surgeon want to see it, they switch on the fluorescent light and they can see just what is left, and that's what sticks to the cancer. So it's very simple. The first clinical trials we have completed now, it was a very good result. It was as good as we could have get it. We was at phase 1/2A study where we increased the dose constantly until we either did not see any further improvement in the image quality or until we saw side effect. We did not expect seeing any side effect judged by the animal data, and we have seen a very well-tolerated product. And that has allowed us to go up, very high up in dose, where we see a very strong image. And we see it both in the light intensity, but we also see it in the contrast we create. Already from 8 milligram approximately, we have a guidance of the surgeon. And since then, all the patients light up, and we are now up on 48 milligram with the highest dose, and there first we started seeing a decline in the image quality because the background came up and disturbed the image. So 36 milligram in the evening was the optimal dose we had decided going forward with. And the key thing with having a robust result is that we both have across patients. So that means regardless if there's a big and a small, where the cancer is located, we see it very robustly. It's also important because of the equipment used. We have used 4 different equipment in the trial, and even in the poorest equipment, we see very good light on it. And I'll try to show you an example of how it looks. See if this works. Yes, it does. There you see the image during the surgery, and you see the cancer in the middle lighting up very much like a sun, and then you see the rim around where the cancer cell is, and that is what the surgeon used to guide them. And you can see very clearly where the cancer stop and where the normal tissue start. And really, that is a result. But now you can see that the neurosurgeon is kind of dissecting, so separate the cancer from the normal tissue to be able to take out the cancer. So the white bubble in the middle is the cancer, and then the dark tissue around is the brain. And what we are working with is a particular receptor on the cancer called UPAR, and that is a receptor that is the cancer is an enzyme system the cancer used to spread into normal tissue. It's very relevant because it's more expressed on cancers that tend to spread, and it's expressed exactly on the forefront of the cancer, exactly where the surgeon would like to see it. It's a unique feature with it is that it's not limited to one cancer type only. It goes for 80% of all solid cancers, so all the big one, the breast, the colorectal, the lung, and then a lot of small cancers. And that is defining how we go ahead with our pipeline. We have 2 indications we are focusing in on. The one is brain cancer, which is a small indication where we can get quickly to the market. And then we have the lung cancer, which is a larger indication that is actually the most frequent cancer in the world. So for us to be a market leader, there's 3 things that is needed. One thing is that we need a large market, and another thing is we need a unique technology, and then timing. So the market is big. We have a unique technology. We are specific to cancer. We are going across different cancer indications, and we're in a plug-and-play to the equipment. On the timing, there's 3 things that have happened in the surgery. It's becoming smaller, it's becoming digitalized, and it become automated with a surgical robot, and we actually facilitate this process. I mean, the minimal surgery have started a long time ago. The robotic surgery come, and now the acquisition of camera has been started. Olympus and Medtronic acquire cameras, and the equipment is ready for being really taking our guy on board. So the next coming months here, rest of 2022 will be very exciting. We have 2 phase 2 results, one in phase 2A in the brain, and one phase 2B in the brain and 2A in the lung. We started out the production for phase 3, and then we will have a range of different things coming in 23, starting up the phase 3 program. And I think answering why you should invest in FluoGuide, I mean, you can help a lot of patients. 15 million patients every year have cancer. 80% of them need surgery. The founders of FluoGuide, we have a long-term perspective, 10 years or more. We think it will take another rally since the IPO. And we definitely think that FluoGuide could be a market leader in this sector. And then, of course, there are some very interesting value inflating points that may start the next rally coming up in 22. Thank you.
Thank you for your presentation. First question, your first candidate, FG001, is now undergoing parallel phase 2 studies. What does the road ahead look like for them?
Well, I think the interesting thing is that it was safe. And when it's safe, it can go out in any other indications. The UPAR, which is the target for the product, actually is expressed, as I mentioned, in a lot of the different cancers in the body. So for us, it's more a question of showing that we can work across different cancers, but on the same time stay focused. So we selected the brain and the lung as the 2 indications, and it's really the kickstart. It's a license to hunt in other indications, you can say, we have with this result.
You have a big bet on this company. Being the second largest owner as far as I can see. How important is it for you that the CEO have skin in the game?
It's important, but it's balanced. The CEO is not, should not have too much to say, but be motivated. And I think that's exactly the shareholder structure we have now. We have some key larger investors that actually, where we need agreement among us. So I think it's a perfect shareholder structure we have at the moment.
Question from the audience. Do you expect surgeons to be ready to use your technology if you receive positive results in brain cancer?
If they're ready, yeah, absolutely, yes.
I love short answers.
Yes, but yes.
Yeah. In what way can FluoGuide's products be utilized for automation in surgeries?
It's a very interesting question. The automation now, I mean, the robotic surgery is not doing the surgery at the moment, but they are kind of prepared for it. So it's still the physician that maneuver them and steer them. But for them to start becoming automated, they need something to guide them. And that's exactly what we provide them. I mean, the camera acquisition that is taking place is preparing them for this. And now the next one is the dye, which we are one of the leading companies in providing.
You recently presented positive top-line results from your combined phase 1-2 study on FG001. How should we interpret the results, and what can you tell us about the way forward?
Yeah, but it was as good as it could be. I mean, it was safe. It was. We found the dose going forward. We have the very robust illumination of the tumor, both seeing a lot of light and see it. I mean, the contrast, and it was robust across patient and equipment. Couldn't be better.
Questions from the audience. Do you have an ideal partner going forward to commercialize FG001?
Yes, I mean, we work very closely with all the partners that's out there. We don't want to jump into a partner agreement that would kind of destroy the commercial potential for partners later on. So we keep wobbling all of them at the moment to decide at which time we will do what kind of agreement. And I mean, ideally, we would like to sell and keep the cake. So we both would like to, I mean, have multiple indications, multiple partners, multiple geographies, and we're working on models for that.
Next question. As a Danish company, how come you have chosen to be listed in Sweden?
It was because It allows us to attract the institutional investors. I mean, we was told by some of them we was discussing with that we could not list in the First North Copenhagen and attract institutional investors, so that was the way to go. And for us, again, when we talked about shareholder structure before, for us it's very important to have a mix of larger and smaller and owners, and we think we have a really good shareholder structure now. We have some large investors as well that support us. But it was our view for, as I mentioned, we see it in a long term, we see it in 10 years or more, and becoming an real company, we need all that to succeed.
Question from the audience: what competing technology do you see as most important?
There are a couple of companies out there that do competing technologies. There's some not that functional, well-working products that is old and been approved for some time ago, and there's some new products coming into the market. But I think the key thing, I have to run a little bit quickly because of short time here. There's three things that define the unique technology. One thing is that it's targeted specifically to cancer. It has to be used across the different cancer indications, and it have to be plug and play with the equipment. And the last thing I think probably is the most important thing is that you don't have to go out and install a new laser, light source, or new filter, but it just you can use it with equipment that's already there. And that's why our result is so important, because it's robust. It shows that it could work with the poorest equipment, which we, by the way, had to pay for testing because it wouldn't render to us. But it's important for us that we get it proven even on the poorest equipment. So this is, I think, in a nutshell, it.
Another question from the audience: how will you price this compound once approved?
Yes. As high as it could be justified for the clinical benefit we provide. And the discussion we have right now internally is refining the phase 2B design, because we would like to have a benefit documented in the trial. The more benefit we document in the trial, the higher price we can get. But on the other hand, it could be it take a longer time. So that is the trade-off we're working on, to document the highest possible benefit without adding more patient or adding more time.
Last question.
It did not answer it, but the base case is $10,000 is the base case.
Okay.
Yes.
Last question. Do you have plans to move to a larger list?
Yes, we would look at that at some time, but we are not in a rush now.
Thank you very much, Morten Albrechtsen.
Thank you. Thank you for your time.